So it will be really good to have diversified aspect of biomaterial and how those can be utilized for tissue engineering. So thank you, Jyota Madam, to accepting our kind invitation and really appreciate. So over to you, Madam. Please take over the proceedings. You are muted. You are muted.
Okay. Yeah, I'm sorry. So
Yeah.
thank you, sir, for your
Yeah.
kind words, and I hope my screen is visible.
Yes, it is visible. So if you can go to full screen mode, that will be better. F5. So,
Just a minute, sir. Sorry. Yes.
yes, just perfect.
So yeah.
Please go ahead.
So welcome everyone to the session, and I hope that by the end of this session, you will be able to have a general idea about what tissue engineering is, and if you, in future, if you plan to work on tissue engineering, so I hope I would be able to give you the steps in which you should think that where to go while working on tissue engineering. So you might have always heard these two terms together, tissue engineering and regenerative medicine. Regenerative medicine rarely appears, in a standalone case. It is always associated with tissue engineering and is usually called as term TERM, tissue engineering and regenerative medicine. That is because the main aspect of regenerative medicine is actually tissue engineering, and regenerative medicine is a mere application of tissue engineering among many other applications. So that is why regenerative medicine cannot be standalone, and it has to be always associated with tissue engineering. So the first appearance of the term tissue engineering was in 1993, and it was put forward by Robert Langer and Joseph Vacanti. Robert Langer can be considered as father of tissue engineering, and they have described tissue engineering as a new field that applies the principles of biology and engineering to the development of functional substitutes for damaged tissue. So this was a review article, or rather a perspective type of article, in which they have put forward that what tissue engineering means. And later, the same authors in 1999 have published a review article describing that tissue engineering is the design and fabrication of living replacement devices for surgical reconstruction and transplantation. But a more working definition of tissue engineering was later put forward in 2004 by Lawrence Singh, which states that tissue engineering can be defined as the application of biological, chemical, and engineering principles towards the repair, restoration, and regeneration of the injured tissue using biomaterial cells and factors, either alone or in combination. So we can see that tissue engineering is actually 3 decades old, at least 3 decades old procedure. It is more older than that. It has been used for ages, but it has been appreciated in just 3 decades. And it is a highly interdisciplinary subject. It combines biological, chemical, and engineering principles, as well as that in detail that includes the molecular biology, cell biology, biomaterials, or in general material science, and chemistry knowledge, so that tissue engineering can be made successful. Whereas if we want to define regenerative medicine, then it can be defined as a procedure which combines the tissue engineering strategies with other strategies that include cell-based therapy or gene therapy, sometimes immunomodulation. But ultimately, the aim of this regenerative medicine is to induce organ or tissue in regeneration in vivo. That means inside the body. Whereas tissue engineering is usually done, it can be inside the body also, but all these manipulations can usually be done in the lab, that is outside the body, or in vitro. So that is the basic difference of tissue engineering and regenerative medicine. And regenerative medicine is possible only because tissues can be engineered. So why we need this tissue engineering and regenerative medicine? So one of the major uh, reasons for that is organ failure. So we know that this is the statistics, this picture, it shows the statistics of only in India. So in India, annually 5 lakh people wait for an organ to be transplanted. Their organ has become non-functional or dysfunctional, and they wait for the organ, a new organ to be transplanted to in place of the damaged one. So 586 Airbuses can be filled with such people who are waiting for organ transplantation. And by the end of the year, many of them die because no organs are available. So if we look at the organs, so almost 2 lakhs corneal transplantation are required in India. Wherever every year only 50,000 corneas are donated. So 3 out of 4 people, they are permanently visually impaired due to this lack of cornea. Okay. Other than that, 2 lakhs kidneys, 50,000 hearts, 50,000 livers are required as of recent statistics. But the available organs are only 1684 kidneys, 339 hearts, and 708 livers. And you can see the difference between what is required and what is available. So donated organs are not a solution for the replacement of damaged organs. Not only because less organs are available, but also always donated organs are not compatible to the recipient. Sometimes they are rejected by the immune system of the body, so they are just wasted. Okay, and it is very difficult to, even if the organ is available, but it is very difficult to deliver to the place where it is required. It might be some remote location or far away place, and it is not delivered on time. So either the patient dies or the organ gets damaged. So that is why this we have to find an alternative for organ transplantation, and the most simplest way is tissue engineering and regenerative medicine. So which is supposed to create functional tissues and organs which could be implanted in the recipient in place of any organ from the donor. Other than that, two other major applications of this tissue engineering is creation of disease model. Okay, so if you can mimic a tissue in vitro, that is outside the body, then we can also induce certain diseases which is specific to that tissue. And that's how we can understand the etiology, that is how the disease starts or occur, and the progression of the disease, the different stages of the disease from mild to getting serious. So studying of the disease is very important to find the cure of the disease. And always it is not possible to get a patient or a human to study those disease. So usually what is done, animal models are used. But animal model is not a real recapitulation of human system, obviously, because animals have a different physiology as compared to humans. Not only physiology, their habits, especially the food habits, is also very different. And we all know that our food habits actually affects on the disease or our physiology and all those things. So therefore, this tissue engineering can provide as an in vitro model to study a disease etiology and progression. Other than that, drug screening, pharmaceutical drug screening, that is the toxicity and metabolic activity, metabolism of the drug, can also be studied by creating such models, especially by high microfluidic chip-based platforms which are used for high throughput screening of pharmaceutical drugs. So these two things, that is disease model and drug screening, can be used to develop what is called personalized medicine. One of the major application of this disease model and drug screening is the development of a personalized medicine. Developing personalized treatment strategies was one of the important aims of Human Genome Project. Okay, so basically what could be done in that is we can get cells from the patient. We can take directly the affected cells could be taken, or stem cells could be taken that could be differentiated into the affected cells, or some other normal cells could be taken and they could be induced to form a stem cell which could then form the disease cells. Why these steps are required is that because primary affected cells are first not easy to collect and also not easy to propagate as compared to the stem cells. So once you get these primary affected cells or the affected cells derived from the stem cells from the patient, a disease model for that particular disease can be developed, which could lead to some uh, uh, discovery of some novel markers which could help in easy diagnosis of the disease, and obviously drug discovery, or if any other treatment is required, for example cell therapy or gene therapy or whatever it is. And why this personalized medicine is important? Because we all know that we are susceptible to the disease, as well as we have all the persons have different susceptibilities to the disease as well as to the medicines that are given to treat the disease. It is not only dependent upon the age, sex, or the some allied disease like diabetes or something which is there with the person, but two completely healthy individuals could have different susceptibility to the disease as well as to the side effect of the drugs, because of simply because difference in their genetic makeup. So personalized medicine is a very important aspect, and if such models could be developed successfully, it will give us it will give us a way to develop correct treatment strategy for that particular person, which could be different from any other person with the same disease. So these are the few major applications of tissue engineering. Now, what is tissue engineering basically and how it is done? So we have seen the definition of tissue engineering, that so which basically translates to that we have to build the tissue in vitro. Okay, so how that is done? So what is done is that the first requirement in the tissue engineering is obviously the cells that comprises the majority of the tissue. For example, if you want to engineer a liver, you will look for hepatocytes, because hepatocytes are actually the functional cells which does all the functions of the liver. So we can isolate hepatocytes from the donor, okay, and we have to increase the number of the cells, which is done by proliferating or simply what we can call mitotic cell division. Okay, so that the number of the cells is increased. Once we get a meaningful number, then we have to put the cells or seed the cells in a scaffold. And what is a scaffold? So when the cell is being proliferated, that is usually done in two-dimensional plates. But our body, that is the native state of the tissue, is not two-dimensional, it is three-dimensional. So the dimension also affects what the functionality of the cell, or rather what the cell is producing and what it is doing. So we need to provide an environment which is similar to the native environment of the tissue. The first thing which comes is dimension. So to provide three dimension, a support structure is provided, which is known as scaffold. So once you seed the cells into the scaffold, we get a cell-laden construct. This cell-laden construct is then allowed to develop in a bioreactor. And the characteristics of a bioreactor is to provide a controlled environment. Controlled in the sense, in terms of nutrients concentration, that is inlet of the nutrients, outlet of all the byproducts that is released from the cell, the pH, the gas concentrations like that of oxygen and carbon dioxide and all those things. Okay, so all these things can be easily controlled and usually are fixed, that what nutritional requirements are the cells we usually know, and Then what should be the pH, the temperature, the gas concentration? These all we know and can be easily controlled. But what extra thing is that could be included in a bioreactor are bioactive factors that helps to increase the functionality of the cells that is in the bioreactor. Those bioactive factor could be any biomolecule, or it could be any growth factor or hormone or whatever it is, but it will activate the cell to function further better. So when in the bioreactor the tissue is finally developed to a stage when where it could be implanted into the patient back, okay, so then it is taken out from the bioreactor and is transplanted back to the patient. So to date, we have developed lots of tissue engineering devices. For example, the bioartificial liver is a very successful tissue engineering device. Lots of bioartificial, commercial artificial liver are available. We have artificial skin grafts, both acellular and cellular. Sometimes, like I have already mentioned in the definition, that all these factors, that is cells, scaffolds, and the bioactive factor, okay, they could be used in combination or individually. So in case of skin, both acellular and cellularized grafts are available, that is with cells and without cells. The without cells are only the scaffolds and maybe with some factor, okay, but no cells are present there. Then we have artificial blood vessels. We have many bone substitutes, for example, the 3D substitutes. We also have very successful injectable bone substitutes and artificial pancreas. So these are the some of the few examples which are now available commercially, which are tissue engineering products. So if we come to the first thing that is usually required in tissue engineering are the cells. So the first question that comes in mind when we talk about cell is that where will we get the cells? From where will we get the cells? So there could be many sources. So first is the autologous sources. In this case, the donor is itself the recipient. So the cells from the donor are harvested, cultured, tissue engineered, and then implanted back to the donor. So in that case, it is known as autologous or autogenic cell source. If the donor is not capable of giving the cells, then another donor could be used. In that case, another donor of the same species would be used. In that case, it is known as allogenic donor. Otherwise, a more better option would be if the donor has a genetically identical person to him or her. That means monozygotic or identical twins. But we know that this occurrence of twins is not that frequent. But if there is, it is much better because they are genetically identical. And in that case, the allogenic donor is known as syngenic donor. And in very rare cases, cells from different species can also be harvested and can be implanted into the donor. In that case, it is known as xenogenic donor. But only getting the cells from the source is not important. We have to proliferate the cells also, as shown in the representative figure for the procedure of tissue engineering. We have to proliferate the cells. Why do we need to proliferate the cells? Because when we implant the cells back into the body, it might not function in the way that we have predicted. So we have to increase the number of the cell into a clinically meaningful number, so that after reimplantation, the cells are able to do its function properly. Okay, so usually for different tissues, different numbers of cells are required. It totally depends on the number of the tissue. So in general, we can say that around 10 to the power 6 to 10 to the power 9 cells per ml of the scaffold is usually required. For chondrocytes, we need around 10 to 100 into 10 to the power 6 per ml. For liver, 10 to the power 10 per ml. And for skin graft, some 10 to the power 7 to 8 ml per ml of cells is required. But there are limitations to arrive at this clinically meaningful number. So we know that every cell, unless and until it has been cancerous, it has converted into a cancer cell. Okay, they have a limited capacity to divide. So basically, human cells, they can, in culture, when you take it out from body and culture it in vitro, it can divide almost 30 to 50 times, and one single cell can produce around 10 to the power 10 to 10 to the power 15 cells in culture. This limitation is imposed by a limit which is known as Hayflick limit, okay, which is because of shortening of a region of the chromosome which is known as telomere. So with every cell division, the telomere, which are present at the ends of the chromosomes, they are shortened, and when they are completely exhausted, then the cells start aging and senescence occur. Okay, so this graph usually shows that how much, how many weeks the cells can actually thrive in vitro. So if you just take out the cell from the body and try to culture it in the in vitro setting, it is known as primary culture, then it goes up to around 2 weeks. If it is cell line, that means the cell, the primary cells has been induced to become immortalized, or to become, to have many unlimited doubling characteristic. Then it is known as cell line. In that case also, it goes up to 12 weeks, after which telomere shortening occurs and the cell starts aging. Otherwise, it can go a cancerous transformation, okay, and it can divide indefinitely, but the cell has now become cancerous and it cannot be reimplanted in the body. So if you look at the number of the cells that is required, it is up to 10 to the power 9 per ml. And a single cell in culture can produce 10 to the power 10 to 15, according to Hayflick limit. So Hayflick limit does not impose any limitation in production of clinically relevant cell numbers. Then what is the problem? The problem is that the rate of cell division for different tissues is different. For example, the hematopoietic progenitor cells, that is the cells which produce the blood cells, different types of blood cells like RBCs, WBCs and all. Okay, so they have a doubling time of 11 to 12 hours, which is fine. Fibroblast, skin fibroblast, they have a doubling time of 15 hours. Chondrocytes, they have 24 to 48 hours. So these things could be managed. Okay, but when you take out the primary cells, always remember that they can grow up to only 2 weeks in the culture. So when you have a 48-hour doubling time for a cell, so number of doublings will be too short in 2 weeks. So you might not arrive to the number that is required to be clinically relevant. Similarly, hepatocytes, that is the liver cells, they have an infinite doubling time. They actually do not proliferate in cell culture system. And the cardiomyocytes cannot be cultured. They just cannot be cultured. So the different ability of the cells to proliferate, that is difference in the doubling time, imposes the major limitation to achieve the clinically relevant cell numbers. And one of the solutions to this could be stem cells. What are stem cells? Stem cells are characterized by their property of self-renewal and their ability to differentiate into different lineages. So basically, self-renewal means they can produce more stem cells and they have infinite capability of doubling, of division, at least for the lifetime of the organism. So this replication potency, that is the infinite capability of the stem cell to divide, and the clonality, that is one stem cell can produce other stem cells, as many other identical stem cells as well. These two properties account for the self-renewal of the stem cells, and the potency, that is the ability of the stem cells to differentiate or convert into the cells of other lineage. That means stem cells are basic types of cells which could be converted into, let's say, bone cells, to liver cells, to blood cells and all. And depending on this differentiation potency, they could be called unipotent, which could be converted to only one type of stem cells. Hello. Yeah. So unipotent stem cells, for example, the hepatic stem cells or the liver stem cells, which could only produce hepatocytes. Okay, or it could be pluripotent stem cells, for example, the mesenchymal stem cells, which could produce bone cells, the cartilage cells, and it could be induced to differentiate into many other cell types as well. Or it could be totipotent, which could produce all the cell types that are present in the body. And the most classic example of that is embryonic stem cell, the stem cells which are produced in embryo, present in embryo. Obviously, that's how the whole fetus is formed. Now, so this problem of the clinically meaningful cell number of could be achieved by differentiating stem cells to the cells that we require. Okay, but other than that, there are other problems associated with the cells, like the implantation of the cells. So how to deliver the cell to the site of injury or damage. So if we just simply inject the cell in the bloodstream, then only 10% of the cells will be grafted at the site of requirement. All others will be removed by macrophages, by phagocytosis, because that is the natural immune system of our body to remove the foreign things. So only 10% of the cells. So if only 10% of 10 to the power 9 cells, if injected, okay, if they are grafted, and we want all the 10 to the power 9 cells to be grafted, so imagine the number of cells that would be required to inject. And obviously there is a limit of dosing, okay, how much things you can inject inside the body, that also has a limit. So it is very important that the cells should be delivered locally to the site where the damage has occurred. And the biomaterials or scaffolds can do that things, can be a solution for the site-specific delivery of the cells. So before we go to the scaffold section, we would also have to consider that they're in tissue, because it is tissue engineering. So in tissue, cells are not the only things that is present. There is another very important component of the tissue which is known as extracellular matrix. Okay. So this extracellular matrix are actually interconnected networks of different macromolecules like polysaccharide, proteins and all. And they have very high diffusivities. Okay, they do not flow away. They stay at their place only. Okay, and they are multimodal. That means they have binding sites for both cells and other ECM molecules. For example, the polysaccharide, they held the proteins held the polysaccharide, and vice versa, and all those things. Also, the cells attach on the ECM. So the ECM is multimodal, and they can bind many different components, which could be either the other components of ECM or the cells. And from where the ECM comes from, it is synthesized from the cell. The cell synthesize the component of the ECM, they secrete it, which gets layered, okay, or are deposited in a specific pattern and orientation, which is also modulated by the cells. So ECM is for the cell, by the cell type of component. Once the ECM is synthesized, now the ECM has the capability to regulate the fate of the cell. For example, it could help the cell adhesion, obviously, because they have moieties for the cell for binding. They could help the cell to spread and get certain shape. They could help the cell to migrate from one place to another. They give the substratum for the cell to migrate from one place to another. They help the cell to differentiate, and they also can induce death in the cell, that is apoptosis. And another important feature of this extracellular matrix is that they change conformation or under the mechanical load. So these are the characteristics of the extracellular matrix. Other than the extracellular matrix, the tissues also have different cells. There are different cells in different tissues, and in the same tissue we have different cells. For example, epithelial tissue could be composed of absorptive cells, ciliated cells, or secretory cells. Okay, and it is present in they are uh, distributed in the organs according to their function. For example, absorptive cells are present in the intestine, stomach, because they that is from where the nutrients are absorbed. The ciliated cells are present in the lungs and trachea. Secretory cells, for example, the glandular cells, the endocrine glands, which produces hormone, or the liver that produces albumin. So different tissues have different cells, and the same tissue can also have different cells. For example, the nerve tissue is composed of 4 major types of cells: neurons, glial cells, Schwann cells, and oligodendrocytes. So there are 4 different types of cells in the nerve tissue. Similarly, different connective tissues have different types of cells. The tendons, they usually have fibroblasts. Bones have osteoblasts. The adipose tissue have adipose cells or fat cells. Cartilage have chondrocytes. And in all these tissues, epithelial, nerves, or connective tissue, there is another tissue, that is the vascular tissue, or the arteries and veins which carry blood, which is composed of endothelial cells. So a tissue is a combination of different types of cells. So based on these different aspects of this tissue, a microenvironment for the cell is created, which is characterized by the neighboring cells that is present in this tissue. It could be the same cell or any other different type of cell, homozygous interaction or heterozygous interaction, both. Then we have ECM. We could have many ECM components or chemokine, cytokine, which are released from the immune cells. We could have hormone in that tissue. Okay, the gas concentration, that is the dynamics of respiration, oxygen or carbon dioxide concentration could be low or high in different tissue, and the provision of the nutrients and removal of waste byproducts, okay, which are mediated by the bloodstream. So all of this constitute a microenvironment for the cell, and this microenvironment is very, very important for the function of the cells, because suppose the circulatory system, okay, if the waste byproducts is not cleared properly by the circulatory system, then it could be toxic. It could generate a toxic stress to the cell, and cell could become dysfunctional. Or if proper oxygen is not provided to the cell by the circulatory system, then also the cell could undergo in a hypoxic condition. If by chance some high concentration of oxygen is delivered to the cell, then it will undergo a oxidative stress. So also the position of the cells is also very important. For example, the metabolically active cells, okay, they should be within the few microns from the capillary, because they are the metabolically active cells are the cells which are responsible in uptake of the nutrients that we have from food. Okay, so these capillaries, they connect the microenvironment of the cell to the environment, the general environment of the body. So the cell microenvironment, what I want to impress here is that the cell microenvironment is very, very important for the functionality of the cell. And while engineering the tissue, we should always keep in mind that what is the microenvironment of the cell. And this altogether poses the major limitation or challenge in implementation of a tissue engineered structure. So accurately mimicking these dynamics of the cell and the microenvironment, that is the change in the oxygen concentration, let's say CO2 concentration, or the rate at which the nutrients are provided by the blood and the waste are removed. Okay, and the microenvironment, that is what different types of cells are present, what different ECM components are present, what is the characteristic of the ECM component. All this poses a major challenge in recapitulating the environment of the cell and therefore the tissue engineering. Other than that, it is also very difficult to scale up all these things. It it might be we might be able to mimic these dynamics and microenvironment in a small setup, let's say a multi-well plate. But to generate a scaled up feature for the same, which could be implanted in the body, would be very difficult. Automated system creation is another challenge, and obviously the implementation of the automated device. So all these things poses the challenge in creating an a tissue engineered device. So the ECM component of the cell microenvironment could be solved by providing a scaffold. So as I have already told that scaffolds are usually three-dimensional support structure which are designed to facilitate the cellular growth and proliferation. So while synthesizing a scaffold, we should always focus on the selection of the materials to be used. For example, for liver and bone, the scaffold from the same material would not do good to both of them because the extracellular matrix of liver and bone are completely different. So we should, while selecting the material for the extracellular, so for the scaffold, we should always check what is the microenvironment of the cell, especially the ECM components. Then the fabrication method that should be used to produce the scaffold, because different fabrication method of the same material could generate different physical properties of the scaffold, and the physical properties also has a major role in the function of the cell. Other than that, the architecture, the surface properties, as well as the mechanochemical properties of the scaffolds are also very important. Okay, and overall, the scaffold, all these properties of the scaffold can very much influence the attachment of the cell, the proliferation, differentiation, and migration of the cell. In total, all the cell fate processes could be controlled by the scaffold and therefore the functionality of the cells. So, what are the key issues that we usually get in designing scaffold? So first is the surface property. Obviously, the surface chemistry, charge, morphology, and area. All of them are very important for the cell. The surface chemistry usually defines whether the cell could adhere on the scaffold or not. So there are not much cell in our body which could grow in suspension. Okay, most of the cells, they require a substratum for adhesion, and then only they can grow. So this surface chemistry actually decides that whether the cells can adhere or not. The charge on the substrate surface can also be a important characteristics, because the charge would actually dictate that what components can be deposited on the scaffold. It could control the components that could be deposited on the scaffold. And for example, in the tissues, for example, nervous tissue, the charge could play a very important role. It could help in signal transduction and all. And then the morphology of the substrate. Okay, if a rough surface usually allows for better cell adhesion as compared to a plain surface. Roughness in the micro roughness, which could be studied by AFM, like atomic force microscopy. Okay, by naked eye everything appears plain, but I am talking about the micromorphology or micro roughness of the surface of the scaffold. Then the surface area, because more is the surface area, there is more place for attachment of the cell, more place for the cell to grow. Then the architecture of the scaffold, like porosity, pore size distribution, pore volume, and the interconnectivity of the pore. All these are very important because these things actually dictates that what will be the rate of incoming of the nutrients and outgoing of the waste byproducts by the cells. So interconnectivity, porosity, all these things are very important. Sometimes the pore volume and the porosity also provide cells uh, space for the cell to grow. Okay, so what happens is that usually the cells, they can either grow as a layer in a 2D layer on the 3D scaffolds, on the 3D surface, but in a single layer. But many cells, for example, the liver cells, they prefer to form a bunch of the cell or spheroids. Okay, so for these spheroids, formation of these spheroids, the pore volume, okay, and the pore size is very important. For example, this in this SEM image, which is a SEM image of some scaffold, okay, the cell, a hepatocyte would grow on this big pore, very nicely on this big pore, because there is so much area. Okay, so there won't be much contact inhibition, not with other cells, not with the wall of the scaffold, as compared to the small spores. So the pore volume and the pore size can also be important for different cells. And then the mechanochemical properties, the stress strain relationship, the Young's modulus, the compressive modulus, the strength of the scaffold. It is very important. For bone, obviously we need a very high strength scaffold. For, let's say, fat cells, we need a soft scaffold. In fact, the compressive moduli or the strength of the scaffold also can sometimes also define the disease stage of the scaffold, especially in case of liver. So the liver, whenever the liver gets diseased, okay, it usually undergoes a huge modulation in its ECM component deposition. Okay, so a normal liver, they usually is very soft, have a very oriented and very organized ECM collagen. Okay, but as the liver gets diseased, okay, the softness decreases and the moduli, compressive moduli of liver increases. So the next stage in the liver disease from normal to a disease state is fibrosis, after which it becomes cirrhosis. So the normal fibrosis and cirrhotic liver can be distinguished from each other solely on the compressive moduli, based on the compressive moduli of the liver. Okay, and even it is very interesting that if you design a scaffold which mimics this compressive moduli of the different stages of liver disease, and you try to grow hepatocytes on that, you will find the hepatocytes are more functional if they are grown on the scaffold that mimics the compressive moduli of normal liver. And the functionality decreases as you go to the fibrotic and cirrhotic compressive moduli. But if you want to differentiate the stem cells into hepatocytes, then a fibrotic and cirrhotic compressive moduli will be more helpful, because for the liver stem cells, the increase in the compressive moduli is actually a signal for the stem cell to differentiate into hepatocyte and rescue the organ from the disease. So this is such an interesting phenomena which occurs in the liver cells, that within the range of few, let's say 2 to 20 kilopascals, you could get different cell functionality. For hepatocytes, the lower is the compressive moduli, it is better. For stem cell differentiation into hepatocyte, the higher is the compressive moduli, it is better. Then the degradation rate of the scaffold is also important, especially when it is used for regenerative medicine, that is when you implant it back into the body. Because the degradation time of the scaffold should be optimum. You don't want the scaffold to stay in the body forever, because tissue engineering aims to restore and regenerate the tissue and not to replace it. So you want your scaffold to disappear, and the cells that has been delivered with the scaffold will regenerate the organ eventually. So the rate of regeneration of the organ or the tissue should coincide with the rate of degradation of the scaffold. If the scaffold degrades at a faster rate, obviously the benefits provided by the scaffolds will be lost and the tissue might not be completely regenerated. If the scaffold stays for a very longer time, then also it can hinder regeneration because it would not provide the appropriate space for the tissue to get regenerated in time. So all these properties should be kept in mind while designing a scaffold. And for each tissue, there are different properties. So each tissue needs a different scaffold and they impose the issues which are there in the scaffold. So basically an ideal scaffold should have sufficient porosity for the migration of the cell, for the diffusion, exchange of the nutrients and the gases and the wastes. Okay, it should also have the adequate mechanical stability and the surface chemistry and microarchitecture. So based on these ideal properties, we could say that a scaffold should have these characteristics, that is high porosity, which will allow for seeding of the cell and the migration of the cell, pore size that will affect the growth of the cell, internal surface area also, which will affect the attachment of the cell. Okay, and the porosity also accounts for the exchange of the nutrients and the gases and wastes. Okay, then a high surface area so that we could get a clinically meaningful number of cells which is relevant for implantation. Then structural strength, which completely depends on the tissue that is being engineered, and specific 3D structure. Okay, sometimes for especially in case of blood vessels, the nerves and the bones, we have to design the tissue in the exact shape in which the damage has occurred. For the nerves and the for the vascular tissue, we need to construct a tube which would connect the damage in the vascular tissue. For the nerve, some aligned channels should be there in the scaffold so that the nerve cells does not grow randomly and can grow in an organized manner. If there is a fracture in the bone, then the scaffold should exactly fit at the fracture site so that excess bone formation and all those things, all those complications are not created. So these are the different characteristics which should be kept in mind for designing scaffold. Then what are the different types of scaffolds that could be synthesized? So I'm sure in the previous session you must have been introduced that there are many types of biomaterials that are present. So, but the function of biomaterials can be many different. It could have myriad of function, not only scaffold. But usually as a scaffold, polymeric or ceramic materials or the composites of the both are used. Why not metallic? Because metal scaffolds can be used for replacement, but tissue engineering usually deals with the regeneration and restoration of the tissue function. So the metallic scaffolds are usually avoided. Yes, sometimes it could be used for drug delivery and can be a part of regenerative medicine. But usually for the scaffold synthesis, polymeric and ceramic are the more better choices. So polymeric scaffolds can be synthesized from natural polymers, which are derived from the natural sources, or synthetic polymers, which are chemically synthesized. Ceramic scaffolds are mainly calcium phosphate or bioglass or biosilicate. Those types of scaffolds are used. And composites, they are those scaffolds which are usually a mixture of both of them. Okay, polymer and ceramic. If two different polymers are used or two different types of ceramic are used, they are usually called blends. But composite are two different classes of biomaterial which are used to create a scaffold or something. So there are many fabrication techniques also. I have only listed the three major techniques which are now used to fabricate the scaffolds. So one is the hydrogelation. So in hydrogelation, the gel precursors, which could be a polymer or it could be a monomer also in case of the synthetic scaffolds. Okay, they are mixed together and allowed to react and cross-linked. The cross-linking could be photo cross-linking, a physical gelation, or a chemical cross-linking. And the cross-linked polymers that are they form a gel. Okay, and if you look at the same image, they have this kind of architecture which has very nice and interconnected pores we can see here. So this is basically hydrogel. If the hydrogel is synthesized at sub-zero temperature, then it is cryogel. In case of cryogel, the ice crystals that are formed during the polymerization process, they act as a pore forming agent. The pores are usually result for that ice crystals of the solvent. And then we have freeze drying, in which obviously this is the next step of cryogelation. Okay, in this also the hydrogel is allowed to react, then it is frozen, and directly the ice is evaporated or sublimated into the gaseous phase, leaving behind large pores. So in cryogel and freeze drying, the porogen is usually the ice. In hydrogel, there is no porogen used. It is just the simple gel formation at the room temperature. Then another famous technique is electrospinning, in which the polymer solution is deposited either on a rotating mandrel or on a plate, horizontal plate, vertical or horizontal, whatever, 2D plate. Okay, it is done under electric field, and that's why the polymers form aligned fibers, and they are very useful in formation of sheet, and this is how an electrospun fiber or sheet looks like. Okay, and a more recent and advanced technique is the 3D printing. So usually for electrospinning and hydrogel can form composites. Okay, but it is very difficult to form composite structure in electrospinning. But 3D printing is a very important technique which can be used to form polymeric scaffolds, the ceramic scaffolds, as well as composites. Okay, so through a nozzle, by pressure and by computer assisted designing, a 3D structure is printed, and the ink could be anything. It could be a polymer, it could be a ceramic, and also it could be cell. Cells could also be deposited by 3D printing. So direct printing of the cell, that means you don't have to seed you don't have to synthesize scaffold separately and then seed the cells. So direct cell laden scaffold could be created by this 3D printing procedure, which is very interesting. And you see that the whole organ can be printed. Okay, and this is a vertebral disc that has been printed using some ceramic material. So 3D printing is the most recent and the most fastly developing process that is used for the synthesis of tissue engineered structures. Then the third thing is that bioreactor part. Okay, so in the bioreactor part, what I told was that we maintain the conditions which are appropriate for the growth of the cells. That could be pH, temperature, the gas concentration, nutrient concentration, and all of those things. And some bioactive factors, especially growth factors that are added. So growth factors could be very essential in tissue engineering approach, because they improve the functionality of the cells, and the growth factors are also an important part of the microenvironment of the cells. Okay, so what are growth factors? So growth factors are simply proteins which are synthesized by the cells. They act locally and they affect almost all the cell fate processes, like the proliferation, migration, differentiation, apoptosis, and everything. Okay, so based on the action of the growth factors, they are usually classified as autocrine or paracrine. In case of autocrine growth factors, the growth factors affects the same cell which has synthesized it, whereas in paracrine effect, the growth factors they exert their effect on the neighboring cells. Okay, so this is a graph that shows the hypothetical timeline of the cell fate processes which could be affected by a growth factor. So within 1 or 2 hours, they could influence the adhesion of the cell. Within few hours, they could influence the shape and migration pattern of the cell. Okay, in almost a day, they could help the cell to replicate its DNA and prepare for division. And several days, they could help in proliferation and differentiation of the cell. So this hypothetical timeline is also important because this gives you an idea that up to what duration you could treat the cells with the growth factor, depending on what you want from the cell. If you just want the cell to adhere, you could just put the growth factors for few time. If you want the cell to divide, proliferate, or differentiate, you have to maybe add the growth factors in the bioreactor for many days and all those things. So there are a myriad of growth factors which are present in the body. Okay, so most common ones are insulin growth factors, fibroblast growth factor, TGF beta, BMPs. Nowadays, BMPs are considered as a type of TGF beta growth factor only. So this BMP is the most studied type of growth factor in tissue engineering. So the bone morphogenetic protein, it seems like they are a part of bone formation or something, but it is not true. They are not only the part of bone formation, but in the embryo they take part in development, every developmental stage of embryo, and almost every, for the development of every organ, these BMPs are required. They are called bone morphogenetic proteins because first they were discovered to induce ectopic bone formation. That means bone formation at a site where usually bone is not present, let's say the skin. Okay, they can form bone and skin. That's why they are known as bone morphogenetic proteins. But apart from bone, they are also important in growth of many other organs. For example, this BMP4, they are also important for lungs and eye development. There are many types of BMPs which have different functions. BMP2, 3, and 4 are the most common. So BMP2 helps in osteoinduction, osteoblast differentiation, and apoptosis. That is all the activities associated with bone formation. Whereas BMP3, it is most abundant in bone, but it inhibits osteogenesis. So the shape of the bone does not overgrown, is actually controlled by the activity of this BMP3. And then we have BMP4, which also have osteoinductive properties and required for other organs as well. So in many of the further examples, I might take BMP, the delivery of BMP, okay, as BMP as a growth factor for the study, because it is the most extensively studied growth factor to date. So growth factors can be delivered in different ways. So first, it could be delivered as a DNA. Okay, the DNA could be transcribed and then translated to the protein or the growth factor. This is known as gene therapy. Or the DNA could be incorporated in the cell and the cell could be transferred to the site where it is required. This is known as cell therapy. Otherwise, the protein or the growth factor itself could be incorporated in a carrier molecule or a vector molecule, and that carrier molecule could be transferred to the site of the delivery. That is known as protein therapy. So in case of gene therapy, okay, for example, again we come to BMP4. So suppose we want to deliver, want expression of BMP4 growth factor at a certain site. So how to do that? So what could be done is that genes could be gene for BMP4, the gene, the DNA that produces BMP4 could be introduced in a donor. The donor could be biological, not a donor, but host, okay, or a carrier molecule, sorry. Okay, so the carrier or vector could be biological, that means a virus, or it could be chemical also, for example, liposomes and all. And it is manipulated so that it merges with the cell and the gene is introduced inside the cell. The gene can later enter the nucleus and get incorporated in the chromosome. Okay, or it can replace the defective gene which is in the chromosome, and the functional protein could be produced. So the delivery of the gene via viral or non-viral vector to the affected cells is known as basically gene therapy. Now, there are two ways in which gene therapy could be done. It is either in vivo, in which directly the therapeutic gene is incorporated into the vector and is introduced in the patient. It is a less invasive procedure, okay, but it is very difficult to deliver it to the target uh, uh, site, as well as it could elicit immune response. Then the other method is ex vivo. In ex vivo, the cells are first removed from the patient. The gene is introduced in the cell in an in vitro setting. And the modified cells, that is the cells which has received the gene, then they are implanted back into the patient. So since the cells that are taken from the patient is actually the target cells, okay, which had that faulty gene, so targeting the cell is not a difficult process. So it is already targeted. And obviously it is the patient cells which are introduced back into the body. So immune reactions are always very minimized. But it is an invasive procedure. Okay, and it requires a lot of time as well. And this ex vivo procedure of gene delivery is actually cell therapy. That is the second type of gene delivery, growth factor delivery. Now, there is a very interesting commercially available cell therapy nowadays to treat cancer, which is known as this Carte cell therapy. So, CAR is a man-made receptor, which stands for chimeric antigen receptor. So, usually the cancer cells, when we get cancer, cancer cells are cells from our body itself. So, the immune cells, especially the T cells, they don't recognize the cancer cells as foreign cells, and therefore they do not uh, take any step to destroy those cells. Okay, but if the T cells could be modified in such a way that they could recognize that cancer as a foreign cell, then they could destroy it. And that is exactly what is done in this CAR T cell therapy. So, what is done is that the T cells are harvested from the blood of the patient, okay, and a gene is inserted which expresses this chimeric antigen receptor on the cell surface. Okay, once it starts expressing the receptor, the number is increased to a clinically relevant number, and then it is infused or transmitted back to the patient. Now these cells could recognize the cancer cell as foreign body, and this will destroy the foreign body. The cancer cells, and that is how it could create cancer. This is an FDA approved cell therapy approach, and now many commercial companies are also making this CAR T cell therapy. And for different cancer types, different chimeric antigen receptors are there. So based on the cancer, different CAR T cells could be produced, could be implanted back into the patient. So the next is protein therapy. So the first two methods, this gene therapy and cell therapy, the success rate is actually very low. It is very difficult to manipulate the gene properly. Okay, so protein therapy is much more simpler and a more successful approach. So growth factors, since they act locally, so they must be delivered directly to the site where it is, target site where it is required. For that, we need a carrier. Okay, so the carrier must support the controlled release of the growth factor, so that the rapid clearance of the factor is not done, and it is delivered in a predictable manner, and there is time for the growth factor to show the effect. Okay, and one of the things that could be achieved by that is scaffolds. Again, we come back to the scaffolds. Okay, the scaffolds, since we are taking here an example of BMP, so The scaffold properties, obviously, it imparts a challenge here. So the bone cells must be able to migrate and grow inside the scaffold, and also the scaffold should allow the endothelial cells to come and migrate, because it is very important for the formation of blood vessels or angiogenesis to happen at the scaffold, so that gaseous and nutrients exchange could take place properly. In addition, the scaffold should also justify the controlled release of the growth factor. So for development of scaffold for protein therapy has more challenges, because optimal scaffolding properties for the delivery of protein or a growth factor is much more difficult than to mimic the microenvironment of the tissue, because it is just, a protein that could uh, uh, just get released from the scaffold. Okay, usually the scaffold have high porosity, so obviously the protein retaining capacity of the scaffold could be very low because of the porosity. The porosity will lead to release of the protein. Then hydrophilic materials are preferred for cell attachment, but most of the growth factors are hydrophobic, so it is difficult to retain. So all these things are actually pose more challenge to develop a scaffold which could act as a carrier for the growth factors. Still, many scaffolds have been created or fabricated for delivery of the BMPs for bone growth. Okay, some inorganic materials, for example, hydroxyapatite, tricalcium phosphate, those have been used to create the scaffold. Okay, so they have the advantage that their structure is similar to that of bone. The bone also contains lots of calcium and calcium phosphates and all those things. Okay, but they are usually brittle and they are difficult to mold. Okay, then we have synthetic polymers. Synthetic polymers, we can get a very good control in synthesizing polymers when we synthesize a polymer from its monomer. Okay, because we can always have a control on the synthetic procedure for which is used for synthesis, and therefore we can control the properties of the polymer. But obviously these synthetic polymers are not native to the body, and they usually use harsh solvents for synthesis, and also the when they are degraded, the monomers or the products, the breakdown products, they could be toxic to the body. Then we have naturally derived polymers. Naturally derived polymers are excellent biocompatibility, but they could have affinity towards the growth factor. They could bind the growth factor so that the growth factor is not released. So the delivery of growth factor will be hindered. And they also pose a threat of pathogen transmission. And finally, like all other scaffolds, we also have some composite material which could be synthesized from any of these, okay, combination of any of these. So that could also be used as carrier trays. Now, there are many factors that influence the release of the growth factor. So first is that the affinity of the factor to the carrier. If the growth factor has affinity to bind the carrier, then it might not be released at all. Okay, it could just be wasted. That this affinity could be influenced by the charge, the hydrophobic interaction, and the chemical binding groups which are present on the carrier. Then the geometry of the carrier, that is the uh, uh, pore volume, the interconnectivity of the pore, and all these can actually influence the diffusion of the growth factor from the carrier. Then how the carrier is released. If the carrier is released simply by diffusion, or it is released by degradation of the carrier. So if it is simply by diffusion, the release kinetics might be quite fast and will depend on the porosity and all those things. But if it is on the degradation of the carrier, if the growth factor is released as the carrier is being degraded, then it simply depends on the enzymatic activity that is present in the target site, as well as other cellular activities which helps in degradation of the carrier. And in that case, the release could be retained for a longer period of time, comparatively longer period of time. and also the local host environment. That is, what is the vascularity, the cells, the pH, the ions, all of them could also influence the diffusion as well as degradation of the carrier, and that's why they also affect the release of the growth factors. Then the dose, that how much growth factor is usually need to be delivered, is also important. Okay, usually we supraphysiological doses are required. That means the amount that is required when we are delivering a growth factor is much higher than that which is present in our body. That is because usually this growth factor do not act alone. They act in a cocktail of other growth factors, and presence of other growth factors influence the activity of the of our of the growth factor of our interest. So when the growth factor is acting alone, it may be required at a higher concentration to exert the same effect, okay, which it could exert in a comparatively smaller concentration in a cocktail. Then the delivery system itself poses a much limitation, and that's why higher doses are required. Because, for example, the clearance rate, if the clearance rate is very high, then obviously we have to provide a high dose of growth factor so that it is delivered in appropriate amount which could be functional. And sometimes the action of these growth factors could be confronted by some other factors like the medications. Okay, if some chemotherapy you take, or any other side effects of some other medicines that the patient is taking, or the patient is affected by some other disease like diabetes, the age, the sex, everything could affect the requirement of the dose of the growth factors. Okay, then obviously species to species the growth dose requirements are also varies. We usually test the dose in rats or in rabbits or primates, but we have to use it in humans. So when using it in humans, it could be a completely different dose that is required. So all these factors actually governs that what dose is required for the growth factor, and we should keep in mind while delivering these growth factors. So this is actually all the things that that is used to engineer a tissue. So the cells, the scaffolds, and the growth factor. So we have covered all three of them. Now I will move on to the sustainable strategies for tissue engineering and regenerative medicine. So the sustainability in terms of environmental sustainability, it can be, in case of tissue engineering, it can be achieved only in respect of scaffolds, because the cells and growth factors, okay, it are to be they are to be achieved from the biological sources only. No other source can give us cells, no other source can give us growth factors. Okay, so it is only the scaffold that could be sourced, the material for the scaffolds that could be sourced from different natural and synthetic ways, and sustainable strategies could be applied therefore there only. So what does sustainable strategies take care? So they usually takes under consideration the value addition of the bio waste or any byproduct, not only bio waste, any type of waste or byproduct that is produced in a system, and obviously without neglecting the environmental sustainability, that is for the extraction of those materials from this bio waste or byproduct should also be environmentally friendly. It should not use any harsh chemicals or solvents. So that is also what we should keep in mind, the extraction and fabrication procedure, not only the source of the material. Okay, but there are many challenges whenever we source the material from sustainable sources. For example, the reproducibility of the material could be very difficult, because in nature there could be batch to batch variation. If we get cellulose from one plant, the cellulose from other plant could be completely of different properties. Okay, the porosity, the stability, and tailoring the property becomes a big challenge actually. Whenever the material is sourced from any sustainable place. But still, lots and lots of work are being done in this direction, and these are, yeah, just an overview of what materials that could be that could have a sustainable source and where it could be used. For example, for bone, we could get hydroxyapatite, alginate, calcium phosphate, and dextran. Okay, from the sustainable sources, and we can do bone tissue engineering out of that. Hydrogel, composites, fiber, membrane, all could be produced from all these things. For skin, we could have collagen, cellulose, pectin, hyaluronic acid, zeolite, and chitosan. For cardiac muscles, also we can have cellulose and collagens. And similar to bone, the dental implants can also be formed out of hydroxyapatite and some other important ECM polymers. Okay. So first, I will focus on the scaffolds which has been fabricated by plant-derived biomaterials. Okay, so this is a list of the waste derived material and what are the major findings and where it has been used. So you will see that most of these sustainable sources has been used for bone tissue engineering, because modeling of bone is one of the least intricate. I would say that bone is one of the least intricate tissue. Even if you don't recapitulate the tissue, if you implant it, the bone could remodel the scaffold into its native structure, whereas it is difficult for other organs. Okay, so bone tissue engineering has been very successful and has been done for every sustainable sources, but there are some other organs also, or tissues also, that has been engineered using the sustainable sources. For example, the decellularized scaffold from spinach and parsley leaf, they have been used for cardiovascular tissue engineering. Okay, then apple core and peel, corn silk, they have been used for decellularized scaffold and nanocomposite. This sugar beet waste, out of this sugar beet waste, first bacterial cellulose was used. The sugar beet waste was used as a medium for the production of bacterial cellulose, and then a nanofiber gel was created, which was used as a scaffold. Then jackfruit peel, pectin from jackfruit peel was used to produce a composite, a nanocomposite. Nanohydroxyapatite was mixed with this pectin to produce the scaffold. Okay, and it was used for bone tissue engineering. Then lemons peels were used for skin tissue engineering. Apple pomace was used for bone and dental. Then this gardenia jasmine, a genipin, a cross-linker, which is known as genipin, was extracted from gardenia jasmine. It is a chemical cross-linker, covalent cross-linker, but is of natural origin. So it could replace many chemical cross-linker, for example, glutaraldehyde, which are not good for the cells, which have toxic effects on the cells. So this genipin can cross-link proteins as well as polysaccharide. Okay, and is a very safe cross-linker to use. Okay, then other than that, some curcumin as a fixing agent, which is not directly a scaffold, but has been used for other aspects of regenerative medicine. So it was used for fixing the bone. Then green tea leaf polyphenols was extracted, and they have been used for myriad of functions. I will just give you an example, okay, which is a very interesting paper in which the decellularized matrix. Okay, decellularized matrices, they also come under the polymeric scaffold, because usually the ECM, if the cells are removed from the ECM, then whatever is left are macromolecules and usually polymers. That's why they are known as decellularized matrix and come under the polymeric scaffolds. So the authors in this paper, they have used apple, carrot, and celery. Okay, they have decellularized it, and these are the same images, how they look after decellularization. And based on their mechanical properties, especially the moduli, Young's modulus, that is the strength of the scaffold, they have used it for the tissue engineering of different tissues. For example, this apple, the decellularized apple had the least compressive moduli. So that is why it was used for adipose tissue engineering, which is the adipose tissue is a very soft tissue. So it was used for the adipose tissue engineering. And you can see that how the metabolic activity it increases in this apple decellularized scaffold when the pre-adipocytes, that means which are not completely adipocytes now, but at a differentiation stage just before adipocyte, that was cultured on this scaffold. This is the live dead staining. The green color shows live cells, whereas the red color shows the dead cells. And we can say that barely any dead cells are present. Okay, most of the cells are live. And when the pre-adipocytes are induced to differentiate into adipocytes, then we can see that how this oil droplets has been accumulated, which is shown by this oil red O staining. This is the undifferentiated cells, no droplets are here, and this is the differentiated cells, which shows the accumulation of the oil droplets. Similarly, carrot had a compressive moduli which corresponds the bone tissue. So it was used for bone tissue engineering. Okay, this also barely shows any dead cell. And the bone cells which was cultured on the carrot, they showed a very high alkaline phosphatase activity. Alkaline phosphatase is an enzyme which is produced by the bone cells. So a high activity of alkaline phosphatase enzyme actually shows the presence of the bone cells. And the decellularized scaffold from the celery, which had a very high Young's modulus, it was used for tendon tissue engineering. And here also we can see that there are no dead cells present. And look at how beautifully the tendon cells have aligned themselves. Okay, this insert shows the gives you an idea of the polarity. Okay, degree of polarity of the cells, whereas this cells which was cultured on simple tissue culture plate, a two-dimensional tissue culture plate, they are just random in orientation. There was no polarity, but how beautifully these cells have aligned themselves on the decellularized scaffold, celery scaffold. Okay, so that is how a scaffold can actually influence the properties of the cells and can help the cells not only to be functional but also to position themselves in such a way so that they could do their functions. In tendons and ligaments, the cells are actually organized, which helps them to bear the load. Okay. Then the next important sustainable way to achieve scaffolds is that if the scaffold is derived from industrial waste. We produce a lots and lots of industrial waste. Obviously, there are so many industries, and if that could be, the waste could be used for some useful, manufacturing some useful thing, what could be better than that? So not all industrial waste could be used for scaffold manufacturing because most of them are toxic in nature, but still beer baggage, soybean, plastics, tanning leather, fly ash, they all could be used. The soybean can be used to produce hydrogels, which could be used for many different applications, right from engineering to delivery of drug and all. Then plastics could be used to form composite scaffolds for bone tissue engineering, but the problem is that the plastics are not actually degraded, so it's better not to use them for regenerative medicine. So it's better to use them for in vitro models like disease models or something, drug screening models, anything. So tanning leather can give us biocollagen. Collagen is a very important protein which is present in the ECM of almost all organs. So it is a very useful thing that could be obtained from this industrial waste. And then we have fly ash, which could be a source of ceramic scaffolds. Okay, they are usually aluminosilicate and could form alumina and form biocomposite or ceramic scaffolds. So I will also show a case study of this industrial scaffold derived from industrial waste, in which the authors have combined the waste from plastic PET and soybean oil. So PET by glycolysis, they have converted it to BHET and then combined it with soybean oil. Okay, and a polyester was produced. This polyester was molded into a 3D scaffold which looked like this and was used for osteochondral tissue engineering. That means for both bone and cartilage tissue engineering. So it could be used at the joint. Okay, so in very severe cases of arthritis and all, along with the cartilage, the bone also starts to degrade because of the friction that is created between these two bones. Okay, so such type of scaffolds which are used for both osteochondral tissue engineering could be a very good source for the regenerative medicine of such severe cases of arthritis. And we can see that how the cells nicely proliferate in the scaffolds that are created. These ratios actually shows the ratio of BHET to soybean oil that was used to form the polyester, and we can see that 4:1 to 5:1 ratio of BHET to soybean oil actually gives a quite good result. And then the what was done is that the osteocytes and chondrocytes, that is bone cells and cartilage cells, were grown on the scaffold, and it was checked that how is their functionality. So the bone cells, obviously they produce lots of calcium phosphate, and alizarin red is the stain which stains calcium. So you can see that how beautiful red color is produced in this 4:1 and 5:1 scaffold. So that means the bone cells are quite functional and doing good in this scaffold. Similarly, Alcian blue is a stain that stains a ECM component called glycosaminoglycan, which is a polysaccharide. And this glycosaminoglycan is largely produced by the cartilage cells. In fact, the cartilage itself is actually glycosaminoglycans. 95% of the cartilage is glycosaminoglycan and 5% is chondrocyte. Only 5% cells are present in the cartilage. So you see that this blue color actually shows the presence of the glycosaminoglycan. These stains could be extracted after staining, and it could be quantified also. And we can see that 4:1 and 5:1 scaffold are actually beneficial for the bone and the cartilage cells growth respectively. Okay, so a very beautiful scaffold from the industrial waste has been produced, which could be used in the tissue engineering of both bone and cartilage, and can be used in the severe form of arthritis. Then livestock is another sector which produces a lots of waste. Okay, and which could also be used, since livestock is simply based on, most of them are based on biological organisms. Okay, so obviously they could be easily used for producing some biomaterial and used as scaffold. So majority eggshells are produced, and obviously eggshells, they are made of calcium, so they could be used for bone tissue engineering. Then chicken bones and bovine bones, pig bones, all of them could be used to form scaffolds for bone tissue engineering. Chicken feathers, sheep hair, and hooves, the hooves of the cows and all those things, they are good source of keratin, the protein keratin. Keratin can also be used for fabricating scaffold. And rooster combs, this is a very interesting example. The combs of the rooster, they are a very good source of hyaluronic acid, and it is not only used to fabricate scaffold but is highly used in cosmetic industry, because all these anti-aging creams and whatever anti-aging products are there, they mainly contain this hyaluronic acid. And one of the major source of hyaluronic acid recently discovered is this rooster combs. Okay, discovered earlier but recently used, because it obviously poses the ethical issue of animal source. Some people don't prefer that. Anyways, and other than that, the slaughterhouse blood can be used for muscle tissue engineering. So in an approach to use this uh, livestock waste for the production of scaffold, raw chicken feathers were taken from which keratin was uh, extracted, okay, by simple environmental friendly procedure with SDS and all, detergents and all, okay, and then it was electrospun into fibers, keratin fibers. Okay, and on these keratin fibers, stem cells were seeded and differentiated into chondrocytes, that is cartilage cells. Okay, so it is a very nice example of a sustainable cartilage tissue engineering, and you can see that the cartilage has produced a good amount of collagen as well as glycosaminoglycan, both of which are important component of the ECM of the cartilage. Whenever we talk about the cartilage tissue engineering, it is very important to characterize the ECM as compared to the cell, because in cartilage, as I already mentioned, 95% of the cartilage is actually extracellular matrix. Okay, and only 5% of the cartilage is cell. So extracellular matrix characterization is more important when we talk about cartilage. Finally, marine waste. Marine sources also generate lots and lots of waste, more than we can imagine. In fact, marine sources are one of the major things which produce lots of bio waste. Okay, so they could also be used, but mainly for bone tissue engineering, because there are lots of crustaceans which are present in the sea, and they could be a very good source of calcium and chitosan. Other than that, there are many organisms which produce collagen and all those things. So all those can be used for producing biomaterials. Okay, and that could be used for producing the scaffolds. So in a very unusual approach, keratin, which was derived from tilapia fish, and chitosan. Chitosan are usually obtained from shrimps. Okay, shrimps are a very important source of chitosan. So they were combined to form a scaffold, a sheet of scaffold, okay, in a sheet form, and was used for the engineering of oral mucosa tissue. Okay, so keratinocytes derived from oral mucosa, they were seeded on these keratin chitosan scaffolds and were characterized. So this upper panel, this shows the native oral mucosa, staining of the native oral mucosa tissue, and this lower panel shows the keratinocytes cultured on this fabricated scaffold. And obviously the the cells on the fabricated scaffolds, the morphology is nowhere close to the native, but still you could see that some important markers, for example, the keratin, which is a very important marker for functional keratinocytes, they are present. This brown color indicates the presence of this keratin. Okay, then beta-1 integrin, which is a marker for cell-cell interaction, when the one cell adheres with other cell. So in this mucosa type of tissue, cell-cell adherence is very important because they form a non-penetrative layer, which is mainly facilitated by the cell-cell interaction. So this beta-1 integrin is an important protein, membrane protein, which helps in cell-cell interaction. So this is also present, all these brown things which are there. And then laminin, which is an important component of the extracellular matrix of this mucosa, that is also present, which is shown in this brown color things. Okay, so the completely getting this native structure might be possible at later stages. Okay, this is only cultured for, I guess, a week, as far as I remember. It was only cultured for a week. If you culture for a longer period of time, okay, and with cells, you could get a native morphology. But in vitro, the cells keratinocytes won't grow for that much longer period of time. 2 weeks is the limit for the primary cells to grow in vitro. So if these scaffolds are implanted in the oral mucosa, then obviously it will support this formation of this native structure, because it could thrive for a longer period of time, and there will be other factors also which will help in getting this native structure of this tissue. So we should always remember that waste product or byproduct valorization for tissue engineering or any other application does not limit only to the synthesis of the scaffold, but we should always take care for the extraction of the material, the treatment and purification of the material. If we are doing some chemical modification, for example, most of the materials that are achieved, extracted from the plant, they are polysaccharides. They don't have any cell adhesion motif. Cell cannot actually adhere on that. So we could add some peptides or some other things which would help in cell adhesion. Okay, process optimization and the culture of the cells, everything, all of them should be environmentally sustainable. No harsh chemicals, okay, no harsh solvents, and obviously the source. Everything should be sustainable and environmental friendly, not only the source of the scaffold, but also the fabrication method of the scaffold. So with this, I would like to conclude my presentation, and I would like to first of all thank the organization of the ATAL FDP, especially Dr. Somnath Nandi sir, for inviting me for this lecture, and also my institution, that is Institute of Chemical Technology, Mumbai Marathwada campus, which is in Jalna. Thank you, and I'm now open for questions.
Thank you for insightful lecture presentation. Thank you.
Yes, sir.
Thank you, madam. Really appreciate your time management, and you have given a whole gamut of perspective, and that the diffusion aspects, then how we can able to take the material and no harsh chemical, each and everything. So that clearly indicates the synergies between chemical engineering, biology, biotechnology, as well as mechanical aspect. So that is really good. So actually our target for this sustainability is that in sustainable manner only we can thrive. So you have really given that sustainability aspect in a bigger dimension, how we can hybridize various aspects. So truly appreciate. So it's open for discussion. So any participants, any query, any discussion, please go ahead. So you can ask questions directly, or even you can put it in chat box. So if microphone is not available. So Mr. Alok Raj is telling very informative session.
Thank you, Mr. Raj.
Anuban Deshpande is telling thank you. Uday Kumar, thank you, ma'am. So please, any query, any question, any discussion. So, and another good point is that we had one day for that wastewater and waste removal. So slaughter water and all those, that chicken feather and all those, which are generally, we have considered those as a waste. So nicely you have taken those as the source for biomaterial, and that is the sustainability. That itself is the sustainability.
Yes.
Which is waste. So please, madam.
So either my presentation was very clear, or you have not understood anything. It was not clear at all. So that's why I think there are no questions. Or
madam, this is the general thing for most of the sessions. So very few persons are actually asking, but subsequently there are a couple of questions coming. So we'll definitely pass on to you with the email ID.
Yes, of course, yeah.
And we understand it's because we have tried to I should say hybridize several domains. So that's why the participants are from various disciplines, even from civil, chemical, mechanical, biotech, energy, chemistry, even some people from IoT and computer. So maybe they have different perspective. There are some few queries. So Dr. Srikanth Kulkarni, what are the green and sustainable solvents other than water would you suggest for the synthesis of materials?
Actually, aqueous is the best solvent. For synthesis of any type of tissue engineering scaffold. You can add some salt or some organic solvents like DMSO and all, not in much higher concentration, but in the lower concentrations that would be helpful. But what actually is sustainable is that if you could recover whatever solvents you have used, okay, by some distillation process or anything, even if the harsh solvent is used, okay, if it is recovered and no traces are there released in the environment as well as in the scaffold, because harsh solvents, if they remain in the scaffold, they could be detrimental to the cells as well. So that is also important. But we tissue engineers, we always prefer aqueous solvents because that is the best. Because cells are 70% water. Every living being is 70% water actually. So aqueous is the best thing that we should use.
True, true. And there is a couple of compliments that Tushar Vijay Kadam, he's writing, informative to us. Thank you.
Yes.
Being from electrical background, it is clear to him as well. And
Yeah,
there's
thank
another
you.
comment by Udhaya Kumar. It was clear, ma'am, even we are learning being a mechanical engineer.
Thank you,
So-
Dr. Kadam and Dr. Kumar. And it is really an honor for me that I'm able to build some concept or give a clear idea about tissue engineering.
And actually, that was our target, to at least make them aware about couple of interesting aspects. And it's just so that at latter part of research as well as day-to-day life, how we can able to utilize them in proper manner. So at least if this platform has thrown some light into that aspect, that will also be good.
Yes, yeah.
Uday Shruti T is telling it's very good information to us to gain more knowledge.
Thank you, Dr. Shruti.
Madam, one particular point.
Yes, yes.
I think during some wounds or other things, earlier the stitches were there and now the stitches were different. Soluble stitches are coming. I think
Yes.
you know. So this comes under is same tissue engineering?
No, they come under the aegis of biomaterials actually. They don't engineer tissue.
Okay.
Yeah, so
That's
they are
only a
simple.
soluble
Yeah.
polymer is taken and it is earlier the
Yes, yes.
stitches was is to remove after a particular time period
Yes,
after
both
the
of
wound.
them. Yeah, both of them are biomaterials of different
Okay.
generations.
Okay.
So as there are no further questions, so I should do the formality. So thank you, Dr. Sarkar, for accepting our invitation and delivering such a lovely and very much informative talk, and which has really encouraged many of them. Uh, there is one of the queries just came in. Dhanishta Shagotra is telling, hello, ma'am. It was very informative session. Biomaterials are still very costly when it comes to their use, that people who are not financially stable cannot afford them. What could be done to reduce their cost?
Yes, that is also one of the aim of the sustainable tissue engineering and biomaterials. So if we can derive the biomaterial from a sustainable source, obviously the cost of the source material is reduced, and also we have to focus on the extraction process of the material and the fabrication process of the biomaterial. For example, 3D printing, it requires, especially cell printing, it requires quite a sophisticated setup. Okay, similarly, cryogel production or hydrogel production, sub-zero temperature, it seems like we can put it inside the freezer or something, but it is not easy at that. The temperature should be completely controlled. The heat transfer should be via solvent so that it is uniform and all those things. Okay, so the fabrication methods as well as the source both could affect the cost, and we could work towards or both these aspects and to reduce the cost of the biomaterial and the tissue engineering products.
There is another question from Dr. Srikant Kulkarni. How much is the recovery efficiency of the materials concerned and protocols required for purification of materials?
Yes, I actually cannot give you to be frank, I cannot give you exact figure, but as far as my experience concerned, it is actually difficult. sometimes it is actually difficult. It is just sometimes it is also sometimes lost during the procedure and all those things. So that is why it is more. It will be more better if you could avoid such kinds of solvents and all, because actually the recovery potential is quite low. So many solvents could not be done, very volatile. Many organic solvents are also very volatile, and it is lost in the environment.
Thank you, madam. So definitely we cannot generalize. It depends on the type of process, and it's still not so much matured we can able to provide the cost for this thing. So thank you, madam. Any further queries? Dr. Jain, you want to say something?
Yes, sir. It's excellent presentation, and it give an idea how we can use the waste for our regeneration. Is an excellent concept. Thanks, ma'am, for the nice presentation.
My pleasure.
Thank you, Joyita madam. So we'll definitely the discussion will continue, and we'll pass on the message to you. And if someone